Okay, so the plan is, as we had mentioned before, is that we want to take our first picture, right? This is our very first streak. So we had a little plate, right? So you did your streaking a couple of times towards the center, and hopefully you had some good stuff kind of growing up in there. That's your lab 2. And that's what you're going to submit on Canvas. One picture, that's all I want. If you have extras, that's fine. I just wanted the one done. Now, the idea is that we're going to take this guy and then transfer it onto 2 new destinations. One of them is a slant. It's this guy over here. And then the other one is the plate, this guy over here. All right.
Your other video isn't on.
Right. So
okay.
I'll pop up and I'll switch it off. I'm just trying to use the little screen in front over here.
Oh, okay.
That way we can have some fun. Well, I can have some fun. How about that? So that's the plan. So what we're going to try and do, this is the image that I have over here. You should originally had some nice streaking over here, pretty heavy, and hopefully it got a little bit thinner and then a little bit thinner. And then you should have done a cool little fishtail towards the center. And ideally, you should have had both some white and some yellow, reddish kind of dots. That's the hope. If it didn't work out again, it's okay. Part of the learning process. Now, what we want to do is collect those dots. And then we're going to take, I'm going to darken this up a little bit. Nope, wrong one. Pick up some of those colonies in there, individual ones. And then we're going to use the needle end of the loop. Needle end of the inoculum, I'm sorry. And then we're going to take the slant and do this cool little fishtail upward, which I'll demonstrate using the video. And then we're going to take a second one, a different one. Ideally, it would be the off-white one, but if it's not, you can use a red one again. Just find another one over there. And that one is going to go to the plate. And they're going to do the same process again, kind of the restreaking. Instead of a fishtail, we're changing it up. And back and forth, back and forth, back and forth, back and forth. And then we'll fishtail it towards the center. That's the ultimate goal. Then again, we'll flip the plate. We'll leave the cap loose on the slant, and that's it. That's the overall plan. Okay, so that's what we're aiming for. So now let me turn on the video. So we can, so I can actually demonstrate it over here. There we go. So let's show you where we're going to start. And let me now minimize my presentation so this appears on a bigger screen. All right, everybody should be able to see the same thing I'm seeing right now. So you are going to go back to this guy, right, where you had all your plates and everything else. And so we're going to take out 2 things. We're going to take out one more plate, and we're going to take out this little slant over here that is hanging out over here. So let me take them out. Here's your slant. As you can tell, it has a slant. I know, magical, right? A little slant. And then I'm going to take out one more plate simply because of the fact that these ones look a little bit more dry. And there we go. Second plate. That's all we need. These 2 guys right here. And we're going to need one inoculum. So I'm going to close my plates over here. I'm actually going to have my inoculum already out because of facility for me. Yours should be still sealed. So only take it out once you're ready. But I'm going to just put mine over here to make it easier to start off with. Okay. And so what you should have once you actually take this out, you're going to notice that one side looks like a needle. The other side, if you flip it, has a little loop. The needle side we'll use for the slant, this guy, and the loop side we'll use for the plate. So these 2 guys together and these 2 guys together. That's the plan. Now, ultimately, so these are our 2 destinations. What we need is a source. And our source should have been a plate that looks roughly like this. So sometimes you'll notice Serratia marchescens, which is the one you're seeing here, might have kind of overtook the growth. But you'll notice all 4 streaks, the little fishtail in the center, and then you probably see some individual dots in there. Those are the ones we want to aim for. Now, if you were a little bit luckier, you might end up with something a little bit more like this. And what you see both, a little bit of reddish, a little bit of whitish, off-white, whatever color. And so you want both. You want to use the little needle for the red stuff. You want to use the loop for some of the off-white stuff. Again, if available. If not, just work with the one sample that you have for both experiments. So I'm going to use this guy as my setup, right? I'm going to take this guy to start off. So I'm going to take this one and transfer it to both the slant and the plate. That's the plan. Now, a couple of little quick head substances. I'm going to move this off to the side. If you want to hold your tube or something like that, remember, you can make up your own little holder or something like that. I'm using a little bit of tape over here to make it a little bit easier, right? Or, sorry for the noise in there. You can use, like I said before, the evil trick of my aluminum foil bit, all right? So I'm going to use that for a second. So it's going to get a little noisy, so I apologize. But again, you just kind of crumple it up. Give it the shape that you need. So I'm going to put my slant in there. Bam. Kind of hug it. And then at the bottom, I just kind of flatten it out. So I have to put a little press and voila, magic trick. It stands up on its own. All right. So this is, insanely expensive 1 cent technology at home. Right. Okay. So now idea is take it from the source, this guy, and place it into the slant first. Then take it from the source and then put it into the plate. That's the plan. Now, as always, what I want to do is kind of give you a little bit of a heads up on what we want to do. So by doodling. So let me move all my stuff out of the way. Let me go steal some printer paper. There we go. And I'm going to use a wax pencil over here just so we can kind of do a quick practice. So this time I'm not going to use the shapes because I'm going to be a little lazy. But the idea is you're going to have a plate. Voila, circle. Look at that, perfect circle in my brain. That's many, many years of art school. So again, you're going to divide into quadrants. Right? And the principle again is you're going to streak outside the lines. And so I'm going to move my paper so you can see my hand a little bit more. But the idea is, again, I'm going to try and lay my hand, the blade of my hand, flat on the surface so you can do the technique of just kind of moving back and forth. So I'm actually going to deglove myself really quick on one hand. You shouldn't do that. All right. Here's my hand. And again, flat of my hand, the blade of my hand is flat against the surface. So I can hold my tool over here and only fingers are moving. This is it. Not my wrist. It stays flat. You'll notice that kind of my pinky just stays there, kind of like I'm pretending to drink wine or something like that. And only my fingers move. And so then the idea again is with the inoculum, go back and forth. All right, that's going to give you your awesome, almost straightish lines. Then you go another 90 degrees and then again, and then we rotate it. My paper is a little bit bigger than usual. And again, just back and forth. And then when you make it to the last one, only pluck off from this corner. Do not overlap again from the beginning. So you go towards the center. Bam, little fishtail. All right, that's it. Now you're going to do the same thing for the slant. When you look at the slant, here's my slant. Again, 20 years of art school. When you look at it from the side, you'll see the medium looks like that, a cool little incline. When you look at it heads on, or head on, I should say, it'll look like it has this cool little oval onto it. And so what you're going to do is with the needle, the pointy side of your loop, you're going to bring it down all the way down to the bottom over here at the base of the circle of the oval. Okay. And then from there, you're going to basically paint upwards or outwards by doing a cool little fishtail like this and then go out. That's the idea. Okay. So what we're showing you is this guy. You'll see at the bottom where the little loop appears, you'll touch there and then go all the way up with your needle. That's the plan. Okay. And once you're done with those both, then two things happen. This one, flip. And then this one, leave a loose cap. And I'll show you that in a moment once we do that. That's the plan. So now we're going to take it into effect. Let me put away my little wax pen and get a new glove because I just discarded my other one. All right. So I apologize for the awesome noises. All right. Let me take this guy away. Nobody judge my doodles. And so ultimately, we'll get a source. Again, like I said before, I'm going to use my streak plate over here. I went a little light on this guy. Normally, I do a lot more lines like you saw from the previous plate, simply because of the fact that I wanted to be able to kind of have some clear areas so I can show you. So you shouldn't open yours up. I'm just going to open mine up for demonstration purposes. So leave yours closed. But the idea is you should be able to see the heavy, the slightly lighter, lighter, and then lightest towards the center. That's the help. Now, when you manage these guys and use them, we're going to try and use this so-called clamshell technique. And the idea is that you don't want to pop up the lid open, because now stuff from the air, again, as we'll learn in lecture, starts falling into it, especially as I'm talking, you're contaminating this guy. So the trick is quite simple. Have the lid just pop up just off to the side like a little clam, hence the term clamshell technique. So you just do this. What it looks like off to the side, let me kind of go do this, is you're just lifting a little bit off to the side so you can place the inoculum in without letting stuff fall from the top. Okay, so you'll see me do this, and I'll do this. A little bit of a upward motion right there. Okay, and then with your inoculum, with your cool little tool, then it will fit just fine without letting stuff fall into it. That's the plan. Okay, so let's do this. Let's start off our experiment. The first one is going to be from here, the red colonies that you have, Serratia marchescens in this case, we're going to place it into our slant. That's part one. So let's go off with that guy. Now, again, you will have to use your envelope of 3, take out one of these loops, right? Leave the others 2 back in there nice and sealed. This one you're going to dispose in a moment. Trash is fine. So we're going to use the needle side, right? The pointy end, not the loop end, pointy end, right? And we're just barely, kindly, gently going to pluck off one of the red colonies. Now, you're going to see me do this because I'm going to open it up. You should just kind of do the clamshell thing carefully with some decent, gentle skills. But I just want to show you what I'm actually going to do close up. I'm going to pick a little colony, hopefully somewhere in the middle. I want to get one of the red ones. So I'm letting the camera autofocus really quick. So I'm going to do it very bluntly from one of the heavy ones. I'm just going to do this on purpose. You should be picking up the colonies in the center. But I'm going to just show you. All I'm doing is a very, very gentle tap. I'm not stabbing. I'm not poking. I'm not carving. And so again, my hand is stable. There it goes. It focuses. And it's just a little gentle dab. I'm just going to do this, tap, and that's it. That's all I did. It barely touched the surface, but it did touch it. I'm going to do it again just for demonstration purposes. Just a quick tap. Done. That's it. Okay, I'm not scraping, stabbing, carving, nothing. Don't break the auger. Now, if I zoom this in a little bit more, you'll see where I tapped it. You can actually see a little poke that I did with that guy. All right, so now I take my stuff. You can see a little bit of red on my needle over here. And I'm going to get my slant. Now, the slant, how it works is you have to use your pinky by something called a pinky technique, which is in the lab supplement and the videos. Hold it on with your pinky while I'm still holding on to my instrument. And then you screw or unscrew the cap with the other hand. So the cap is held by my pinky in my dominant hand. Now here's my slant. Okay, I'm going to hold it kind of weird so you guys can see it a little bit better. You're going to introduce your loop, your needle, all the way down to the bottom of it. And then you're going to fishtail quickly. I'm just doing it nice and slow all the way out. That's all we did. So from the bottom, a quick fishtail out. And that's it, done. Okay, you kind of painted the surface of that slant. You return it back to the pinky technique and close it. And reminding you again to leave it loose. Now, what is actually happening here is that you could easily kind of snap this cap on nice and tight. All I want you to do is do this. About a turn to turn and a half. Just do this. And that's it. The cap is loose, but it won't fall off. You know, I can hold it upside down and the cap still remains intact. Okay, so about a turn and a half keeps this cap nice and loose. We've done a fishtail on this guy. And if done correctly, it'll look like this. A little fishtail from bottom to top. And I strongly encourage you to do it quickly. Don't stab it. Remember, it's just painting the surface gently. right? That's the plan. That's what we're hoping for. So we're done with part one. Now we go back to this source plate. I'm going to keep it open for you guys to see. Right? And now I'm going to try and pluck off one of the off-white colonies. Again, if you have one available. If not, still work with the red ones. And now I'm going to transfer that using the loop to a brand new plate. Now, as usual, we're going to want to mark these and everything else. So I'm going to do a quick demo of that again. All right. So this is all the instructions. This is on the supplement, all that good stuff. But one more time, just quadrants, right? I'm doing these really fast, so they're not quite perfect. And remember to label all your good stuff around it. So my name, I would put in my section, my sample. In this case, I would write something like, this is lab number 1, right? And ideally, because this is supposed to have the white colonies in this case, or the off-white colonies, this would be Staph epidermidis. Again, ideally. If not, just work with what you have. I'm good to go. Plate, you can see a little bit of condensation, which doesn't make a difference whatsoever, hence why we flip the plates. And so again, you, following your loop, in good technique, keeping that guy closed, again, I'm opening mine just for demos, you do the clamshell thing, right? So mine, I'm just keeping off so you guys can see it. Same idea, I'm gonna try and pluck off one of the off-white colonies. So I'm gonna just pick up somewhere in here in the center. Again, it's a gentle tap. It's not a carve, it's not a stab directly from above. Again, my hand is nice and stable on the bench. And so let me show you really, really up close what I'm doing. So the camera may do some weird focusing thing. Yep, it's doing it again. Hang on, come on, refocus thingy. There it goes. Just see what I'm doing. I flatten the loop horizontally or parallel with the surface and just a gentle tap. Just bam, that's all I did. You can see I left a little bit of an indent in there. I'm just tapping it. You're picking up literally millions of these guys, and we only need one. Okay, so my loop has the good stuff, and now I'm going to perform what you already know how to do. Now I'm going to, again, clamshell it as you're supposed to do it, just to keep it open. I'm going to leave mine open fully so you can see what I'm doing. And again, same premise, same idea, same stuff. Stable hand. I'm going to put it over here so you guys can see it. And then again, back and forth, back and forth, back and forth a few times, as close to the edge as possible. Rotate it 90 degrees using the same loop. Okay, so you're not changing anything else in this case, right? Same idea, back and forth, back and forth, back and forth. 90 degrees, all right? Keep it nice and flat, back and forth, back and forth, back and forth. And then the last one, 90 degrees. Again, we're gonna pluck off from one side, but not the other. So we go from here and then go towards the center, a few little fishtails. And that's it. Ideally, bam, close that guy. And then we're going to flip it on its bottom, basically. So the top should have the auger and the labels. And then your tube, again, loose cap. And then we're done with these two bad boys. Now the loop, in this case, you're done. You use both the needle and the loop. So you can discard this guy. Now, if you don't put it back into the envelope, you'll contaminate everything. So trash, basically. Since it doesn't really have anything dangerous, you can trash it. You feel a little bit more uneasy, spray it with some Lysol, spray it with some Clorox or something like that. Even alcohol will do the trick. And if you really, really want to be a little bit more sensitive about stuff, take the glove. So I'm going to take my glove off my hand really quick. You won't see it very well because it's tiny. Here's my glove as I'm removing it, right? Shove this guy in there. I'm literally putting the entire needle and the inoculum in the glove. And now you can throw it away. It's nice and sealed and put away. All right. And that's it. This guy can be placed also on its head if you really want to. This one won't really mind if it's kind of on its side, so you don't have to hold it up. But remember, if you constructed your awesome super aluminum thing, that should hold it up too. And so plate upside down, cap loose on this guy, let them sit for about 48 hours. Oops, it's not focusing, and you should be set to go. So now let me go back briefly just to the slide presentation to kind of reiterate the same thing, right? It should pop up on your screen in a second or two, right? And then now, just to recap, you took a picture of your cool streak plate. It's going to show up a little bit smaller on the screen now. Take a picture of this guy and you sent it to me through Canvas. right? And then now you're going to open up that streak that you made, use the needle point, fishtail on the slant. Use again the same source, use the loop side, and transfer the white colonies, if available, and restreak it onto the plate, the big auger plate right there. And that's it. That's the setup. So both transfers, so this is your aseptic technique. This is lab number 1. And then flip the plate so it can grow and not have condensation bother it. And then loosen the cap of the slant. And then that's it. Store them in a nice warm place, away from humans, away from children, away from animals, that kind of thing. Do not put these back in the fridge. Okay, so keep them nice and dry and away from stuff. So that's going to be the plan for this quick experiment at home.